Food safety has always been one of the key issues covered by Marine Nutri-Sciences, and we have to face different hazards in food. That goes on chemical hazards with the contaminants, the foreign bodies that are introduced in the food chain, and the pathogens that are potentially in the food. The emerging new pathogens on the food chain are now really the concern. Salmonella has been here for years, and in the 60s it was the big issue. Now, the virus outbreaks we are facing, and they're international, like the hepatitis A cases, where we start from South America with the production of fruits, the transformation of these fruits in Asia, and the consumption in Australia, where the actual sick people are, because that's where they consume the products, is a typical example. Another one is noroviruses, where the US CDC considers it's #1 in the rank of food concerns because it's over 50% of the sick people because of food. This type of outbreaks really illustrates this field to fork approach, and the prevention measures also have to follow this track. That goes from the raw materials, and here we're talking about products from agriculture. So it goes on water, the quality of the water is critical, and the good agricultural practices have to be evaluated. Then it goes at industry level, and there the quality and the cleanliness of all the equipment is critical, as well as the handling of people and the good hygienic practices. So we can evaluate these, train people, and push for improvement. That's how the viruses will be pushed out of the food chain. Detection of viruses is also critical to help the prevention measures. For years, we only had cell detection of viruses. It was long and not very efficient. Now that there's new technologies like red PCR, we can target the RNA of the virus directly and select norovirus or hepatitis A. We have developed new labs, specific labs for virus detection for that. That doesn't solve another question. Tomorrow, there will be new viruses, new emerging risks. How can we capture and be faster to a solution? The next generation now is called next generation sequencing. We will not only target one virus, but we will cut all the RNA present in the sample, and we'll try to catch all the information. There, we need the bioinformatic treatment of this information, and we're in the world of big data because there is a very huge amount of information to be treated. That will help not only target what we know in the sample, and that's our traditional viruses, but maybe tomorrow's emerging virus that will get in the food we don't know. That's what these technologies will allow us to do, and that's where we go to change our labs for this new holistic approach. The vision we have of the laboratory of the future is to cover from field to fork with a holistic approach that will allow us to cover for all pathogens that are present or might be present, and that's the way we see we can guarantee the safety for all the consumers.